Hello there, and welcome to the Science for Sleep channel. Tonight, we'll explore the vast story of evolution, a journey spanning billions of years where life continuously transformed and adapted across endless generations. You'll discover how whales learned to swim, how humans inherited ancient genes, and how a single ancestor eventually became the millions of species sharing our planet. These quiet wonders have witnessed the slow dance of natural selection, holding secrets we're only beginning to understand. You've got nothing to do but rest. Let your mind relax, your breathing slow, and your body grow light as if gravity itself has softened. Just before we begin, if this helps you rest, consider liking and subscribing. One sleepy soul at a time. Now, let's begin with evolution. All living humans today share a common ancestor with chimpanzees from approximately 6 million years ago. When we look at DNA, the instruction manual for life itself, we find that human and chimpanzee genetic codes differ by only 1 to 2%. This small difference reveals something profound about time and change. Millions of years ago, a population of primates lived in Africa, moving through forests and grasslands. Some of these creatures eventually evolved into the lineage that became chimpanzees, while others took a different path, eventually becoming us. The bones in our hands, the structure of our eyes, even the patterns of our teeth tell this story of shared ancestry. Scientists have discovered fossil remains that bridge this gap, showing intermediate forms between our common ancestor and modern humans. This journey of 6 million years represents an enormous span of time, yet it connects us to every living chimpanzee walking the earth today. When you hold your hand, you're holding proof of this ancient kinship. We are not separate from nature, but rather a continuation of it, shaped by the same forces that shaped all life. Humans carry genetic remnants of ancient viral infections called endogenous retroviruses embedded deep within our DNA. Over 8% of your genetic code consists of these parasitic genetic sequences integrated by viruses millions of years ago. These viral remnants are like fossilized infections frozen in time, creating a geological record of microbial history written in our genes. Long ago, viruses invaded our ancestors' cells, inserting their genetic material into the reproductive cells that would pass DNA forward to future generations. Over countless generations, this foreign genetic material became incorporated into our hereditary code. Today, most of these ancient viruses no longer function, their genes broken and silent, unable to replicate or cause harm, yet they remain within us, harmless passengers carried through the generations like ancient hitchhikers. Scientists study these dormant viral genes to trace human evolutionary history, using them as markers to understand when and how our ancestors lived. The presence of identical viral sequences across all humans reveals our shared past and common origin point. Your body is a living archive of billions of years of interactions between life forms, a testament to the deep history embedded in your very cells. Some of your DNA is shared with Neanderthals, who went extinct 40,000 years ago, proving that your ancestors interbred with a different human species. Non-African humans carry between 1 and 4% Neanderthal DNA in their genomes, evidence of intimate contact between species that seemed so similar yet were distinct. When modern humans migrated out of Africa and encountered Neanderthals in Europe and Asia, they did not simply replace them through competition alone. Instead, they interbred, creating offspring that carried genes from both species into the future. This mixing happened thousands of years ago, yet its effects persist in your body today. Neanderthals possessed larger brains than modern humans, stronger bodies, and different tools and strategies for survival. They had evolved over hundreds of thousands of years in the cold climates of Europe, perfectly adapted to their environment. When modern humans arrived with different technologies and social strategies, Neanderthals gradually disappeared. Yet they did not vanish completely, their genetic legacy flowing through modern populations, influencing how our immune systems work and how our bodies process certain foods. This shared DNA connects us to a distant human cousin, reminding us that evolution moves slowly and that species boundaries are sometimes blurrier than we imagine. Fish developed eyes millions of years ago, and all vertebrate eyes evolved from that ancient fish eye through gradual modification and refinement. The camera-like structure of human eyes, bird eyes, and mammal eyes all traces back to a light-sensing organ in primitive fish from 500 million years ago. Imagine a simple light-sensitive cell in an ancient fish, just capable of distinguishing brightness from darkness. Over generations, some fish with slightly better light detection survived and reproduced more successfully than their neighbors. This tiny advantage, repeated countless times across millions of years, led to the gradual development of more complex eyes. Lens structures formed, protective membranes developed, and neural connections became more sophisticated. Each small improvement offered an advantage in finding food or avoiding predators, driving natural selection forward. The identical basic structure in all vertebrate eyes reveals this shared evolutionary origin, a blueprint preserved across the billions of years since that common ancestor. Whether you are looking at a sunset or reading these words, your eyes are using a design perfected by fish hundreds of millions of years before dinosaurs walked the earth. This ancient gift continues working for you today, proof that evolution's discovered an incredibly successful solution early and refined it through the ages. Your hands contain the same bone structure as a whale's flippers, both having evolved from the pentadactyl or five-fingered limb of an ancient amphibian ancestor approximately 300 million years ago. If you look closely at your hand, you have 5 digits, each with multiple bones arranged in a particular pattern. Look at a whale's flipper, and beneath the skin and blubber, you find 5 corresponding bones arranged in the same fundamental pattern. This similarity in structure, despite radical differences in function, reveals evolutionary history written in bone. An ancient amphibian ancestor, living in swamps and shallow waters 300 million years ago, possessed 5-fingered limbs. Some descendants of this ancestor evolved into reptiles, then birds, with wings, then bats with webbed fingers supporting flight membranes. Others evolved into whales, with flippers adapted for swimming through oceans. Each lineage took the basic 5-digit blueprint and modified it for different purposes, stretching bones, webbing fingers, or adding supportive structures. The similarity in underlying structure beneath such different appearances is called homology, one of the strongest pieces of evidence for evolution. When different creatures inherit the same basic body plan from a common ancestor, but modify it for different environments, it reveals the creative yet constrained nature of evolution. Whales are land mammals that return to the ocean and still breathe air, representing one of evolution's most dramatic transformations across millions of years. Imagine a land-dwelling mammal, similar to modern hippopotamuses, living millions of years ago. Some populations gradually spent more time in water, finding abundant food in oceans and rivers. Across countless generations, Selection favored individuals better adapted to aquatic life, gradually transforming them into marine creatures. Whale fossils show a clear transition from four-legged land creatures to modern whales, documenting this incredible journey. Early whale ancestors possessed hind limbs used for swimming, gradually reduced over time until modern whales retained only vestigial hip bones, serving no function. Their front limbs transformed into flippers. Their nostrils moved to the top of their heads for easier breathing at the surface, and their bodies became streamlined for efficient swimming. Despite these radical transformations, whales retained the fundamental mammalian body plan, including a diaphragm for breathing, warm blood, and the capacity to nurse their young with milk. They must surface to breathe, never developing gills despite spending their entire lives in water. This remarkable transformation took millions of years, yet each intermediate stage was viable and successful in its environment, demonstrating that evolution proceeds through countless small changes, each offering survival advantages. Birds are technically avian dinosaurs that survived the mass extinction 66 million years ago, making dinosaurs not truly extinct but rather transformed into the creatures we see today. When we think of dinosaurs, we imagine extinct monsters of legend, yet every songbird singing at dawn, every eagle soaring through skies, and every chicken scratched in a yard is technically a dinosaur. Theropod dinosaurs, a group of bipedal predators that included Tyrannosaurus rex, evolved feathers and eventually achieved powered flight. These early birds possessed teeth, clawed wings, and other features recognizable as dinosaur characteristics, yet they flew and filled ecological niches unavailable to flightless dinosaurs. When the asteroid struck Earth 66 million years ago, most dinosaurs perished in the catastrophic aftermath of impact, fire, and climate disruption. Yet some of these flying dinosaurs survived, small enough to find shelter and adapt to changed conditions. Over millions of years, their descendants diversified into the incredible variety of modern birds we observe today. From tiny hummingbirds to massive ostriches, from penguins to parrots, all descended from dinosaur ancestors. The genetic evidence confirms this relationship, with modern birds and extinct dinosaurs sharing distinctive skeletal features and genetic similarities. This perspective transforms how we understand extinction and survival, revealing that evolution does not destroy, but rather transforms and reshapes life across the ages. The bacteria in your gut evolved alongside humans for millions of years, creating a partnership so intimate that your survival depends upon them. Your gastrointestinal system hosts trillions of microorganisms, mostly bacteria, that form a complex community called the microbiome. These bacteria descended from ancient lineages that co-evolved with human ancestors, adapting to the specific chemical environment of the human digestive system. They help you digest foods that your own enzymes cannot break down, particularly complex carbohydrates and fiber from plants. In return, humans provide a warm, stable environment with consistent food supply, allowing bacterial populations to thrive. This relationship is so ancient and fundamental that humans and their gut bacteria have become almost a single organism in functional terms. The bacteria produce vitamins like B12 and K that humans cannot synthesize independently, making them essential partners in survival. When humans take antibiotics, these drugs kill not only harmful bacteria, but also the beneficial gut community, sometimes leading to serious illness as harmful organisms move in to fill the empty space. The discovery of this ancient partnership reveals that evolution does not proceed through individual organisms alone, but through complex networks of relationships. We are never truly alone, but rather interconnected with millions of other organisms, all shaped by millions of years of coevolution. Some plants evolved flowers specifically to attract insects, creating a partnership older than 140 million years, with both organisms shaping each other's survival and success. Before flowering plants evolved, the world was dominated by ferns, mosses, and conifers that reproduced through wind-dispersed pollen. These plants invested enormous energy producing massive quantities of pollen, hoping wind currents would carry some to distant female flowers. This strategy was wasteful and unreliable, yet it was the only option available. Then, some plants evolved colorful structures, enticing scents, and sweet nectar designed to attract insects to their flowers. Insects attracted by these rewards inadvertently carried pollen from flower to flower with much greater efficiency than random wind currents. This innovation was so successful that flowering plants diversified explosively, eventually dominating terrestrial ecosystems within tens of millions of years. Simultaneously, insects evolved more specialized mouthparts and behaviors to extract nectar and pollen from flowers, shaping their own evolution around floral resources. Each innovation in one partner selected for counter-innovations in the other, driving rapid coevolution. Today, approximately 3/4 of all flowering plants depend on animals for pollination, most commonly insects, revealing the dominance of this ancient partnership. Antibiotic resistance in bacteria evolves in real time, observable within human lifetimes, demonstrating that natural selection occurs at speeds we can directly witness and measure. When bacteria encounter antibiotics, individual cells with random genetic mutations that confer resistance survive while susceptible cells die. The surviving resistant bacteria reproduce rapidly, passing resistance genes to offspring in what constitutes rapid evolution, unfolding over days or weeks. Penicillin, discovered in the 1920s, was initially a miracle drug against bacterial infections. Yet within decades, resistant bacteria emerged in hospitals and communities. The more extensively antibiotics are used, the more strongly selection favors resistance, accelerating evolution of drug-resistant strains. Methicillin-resistant Staphylococcus aureus, or MRSA, represents one of the most troubling examples, as this bacterium resists multiple antibiotic classes simultaneously. This is not a theoretical evolutionary process, but a practical crisis affecting modern medicine, demonstrating that evolution occurs actively around us. The bacteria do not consciously adapt, but rather genetic variation and natural selection combine to produce rapid evolutionary change. Humans have inadvertently created the conditions for bacteria to evolve resistance, illustrating evolution's power to respond to environmental challenges within time frames measured in human generations. Understanding antibiotic resistance teaches us that evolution is not ancient history, but ongoing process shaping the world around us in real time. The human appendix is a vestigial organ, a remnant from our herbivorous ancestors who needed a larger digestive chamber to process plant matter millions of years ago. Deep within your abdomen, attached to your large intestine, sits a small tube-like structure called the appendix that serves no clear function in modern humans. Your distant ancestors, living in African forests millions of years ago, consumed large quantities of leaves, bark, and plant material requiring extensive digestion. These ancestors possessed a large cecum, a pouch at the beginning of the large intestine, and a substantial appendix attached to it, helping break down tough plant fibers. As human ancestors gradually shifted toward eating more meat, particularly after learning to hunt and cook food, the need for such extensive plant digesting capacity diminished. Over countless generations, natural selection favored individuals with smaller succums and appendixes because the energy saved could be invested in other functions. Yet evolution does not completely discard structures that no longer serve primary functions, if removing them requires significant genetic change. Instead, these organs persist as evolutionary remnants, shrinking and losing their original purpose while remaining embedded in our bodies. The appendix occasionally becomes infected, causing appendicitis, a condition that would have been fatal before modern surgery. Yet it persists because evolution proceeds through gradual modification rather than sudden redesign. This tells us that your body carries a history of your ancestors' diets written in organs no longer fully needed. Giraffes have long necks because their ancestors with longer necks survived better during periods of food scarcity, a process refined across millions of years of environmental challenge. Imagine a giraffe ancestor from millions of years ago possessing a neck of moderate length similar to modern okapi. During droughts, when leaves became scarce and competition for food intensified, giraffes able to reach higher leaves had a significant survival advantage. Individuals with slightly longer necks could access food sources unavailable to shorter-necked competitors, allowing them to survive and reproduce more successfully. Their offspring inherited the genetic basis for longer necks, perpetuating this advantage through generations. Across millions of years, this subtle pressure selected for progressively longer necks as taller individuals consistently outsurvived shorter ones during food shortages. Fossil evidence documents this gradual transformation, revealing giraffe ancestors with progressively longer necks across geological time. The giraffe's long neck did not appear suddenly, but rather emerged through countless small incremental changes, each conferring modest survival advantages. Even today, male giraffes with longer necks achieve higher social status and mate more frequently, continuing the selective pressure favoring neck length. This demonstrates that evolution proceeds through differential survival and reproduction, with individuals better suited to their environment, leaving more offspring who inherit advantageous traits. Darwin's finches in the Galapagos Islands evolved different beak sizes within 50 years, proving that natural selection works on measurable timescales observable within human lifespans. In the 1970s, scientists began studying finches on the Galapagos island of Daphne Major, measuring beak sizes and tracking environmental conditions across decades. In 1977, a severe drought struck the islands, eliminating small seeds that smaller-beaked finches depended upon for food. Larger seeds remained available, but only finches with stronger, larger beaks could crack them open. The finches with smaller beaks starved in disproportionate numbers, while those with larger beaks survived to reproduce. By 1982, just 5 years later, the average beak size in the finch population had measurably increased, a shift that would normally take thousands of years under normal conditions. When rainfall returned and small seeds became abundant again, different selective pressures reversed the trend, favoring smaller beaks once more. Scientists observed this evolutionary process unfolding in real time across just a few human decades. The finches did not consciously adapt, but rather genetic variation in beak size combined with differential survival to produce rapid population change. This research demonstrated that Darwin's theory of natural selection, previously debated as too slow to observe directly, actually operated visibly within measurable timescales under the right environmental conditions. Peppered moths in England evolved darker coloration during industrial pollution, then lightened when pollution decreased, demonstrating that evolution reverses when environmental pressures change direction. In pre-industrial England, peppered moths displayed predominantly light coloration with dark speckles, providing camouflage against lichen-covered tree bark, where they rested during daylight hours. During the Industrial Revolution of the 1800s, factories belched soot and pollution across the English countryside, blackening tree bark and killing the light-colored lichens. In this transformed landscape, light-colored moths became conspicuous against dark bark, making them easy targets for birds, while darker moths blended into the sooty background. Over just a few decades, the moth population shifted dramatically toward dark coloration, a change driven by differential predation, selecting for camouflage matching the altered environment. Scientists collected moths from the same locations decades apart, documenting the color shift with quantitative precision. Then, beginning in the mid-20th century, pollution regulations reduced industrial emissions, allowing lichen to return and tree bark to lighten once more. As the environment changed back, the moth population gradually shifted back toward lighter coloration, proving that evolution follows environmental conditions rather than proceeding in a single direction. This elegant example demonstrates that natural selection responds to current environmental pressures, directing evolution toward adaptive solutions matching present conditions rather than past circumstances. Humans and sea stars share ancient developmental genes, despite 600 million years of separation, revealing genetic conservation across vastly different body plans and lifestyles. Sea stars, also called starfish, possess radial symmetry with 5 arms radiating from a central body, an utterly different architecture from bilateral human bodies with left and right symmetry. Yet when geneticists compare developmental genes controlling body formation in humans and sea stars, they discover remarkable similarity in the genetic sequences and their functions. These shared genes direct basic developmental processes like establishing body axes, forming segments, and positioning major organs in both species. Scientists have even performed remarkable experiments demonstrating this conservation by inserting human developmental genes into fruit flies and observing the formation of functional structures despite the genes coming from a species that diverged billions of years ago. This genetic similarity reveals that successful developmental solutions, once evolved, persist virtually unchanged across enormous evolutionary time spans because they solve fundamental problems of building bodies from single cells. The fact that such distantly related creatures share nearly identical genetic instructions for development indicates these genes originated in an ancient common ancestor, predating the divergence into different animal groups. This tells us that evolution frequently conserves ancient solutions rather than continuously inventing new ones. The deeper we look into genetics, the more we discover that underneath superficial differences, life's diversity rests upon ancient genetic foundations inherited from distant ancestors. Some insects evolved to mimic toxic species they have never encountered through pure genetic accident, demonstrating that natural selection produces remarkable adaptations without conscious intention. The monarch butterfly produces toxins making it poisonous to most bird predators, so it evolved bright orange and black coloration warning predators to avoid it through a learned association. The viceroy butterfly, a completely different species, resembles the monarch so closely that predators frequently mistake it for the poisonous species and leave it alone. This mimicry benefits the viceroy despite not being toxic itself, gaining protection through visual resemblance. Remarkably, the viceroy and monarch do not share close evolutionary ancestry, suggesting their resemblance did not arise from a common ancestor, but rather evolved independently through convergent evolution. Each species arrived at almost identical solutions to similar problems through different evolutionary pathways. The viceroy butterfly populations living in regions without monarchs show less developed mimicry, suggesting that the strength of selection depends on local presence of the model species. This convergent evolution demonstrates that natural selection frequently discovers similar solutions to environmental challenges across different lineages. Evolution lacks foresight or intentionality, yet through random genetic variation and differential survival, it produces outcomes that appear designed with remarkable precision. The viceroy did not evolve to mimic monarchs through conscious effort, but rather through countless generations of slightly better mimics surviving and reproducing more successfully. The peacock's elaborate tail evolved purely for attraction, despite being a survival disadvantage that makes flying difficult and increases predation risk, showing that sexual selection can override natural selection. Male peacocks display fantastically ornate tail feathers in dramatic courtship displays, yet these massive trains serve no purpose in survival and actually reduce fitness in practical terms. Flying becomes more difficult, predators spot the bird more easily, and energy devoted to growing these feathers could support other functions. Yet Paya hens consistently choose males with the largest, most elaborate tail displays for reproduction. This preference for exaggerated traits creates sexual selection pressure that overwhelms natural selection, driving males to evolve ever more extreme tails. This illustrates an important principle in evolution. Natural selection is not the only force shaping life, but rather sexual selection, kin selection, and other mechanisms also drive evolution. A male with a magnificent tail survives less well than a male with a modest tail, but the extravagant male mates far more frequently, leaving more offspring that inherit both the genes for impressive tails and the genes for female preference toward impressive tails. Over generations, this creates an evolutionary arms race where females evolve stronger preferences for exaggerated traits, while males evolve increasingly extreme displays. The peacock's tail demonstrates that evolution produces outcomes that seem wasteful and counterintuitive, following the logic of reproductive success rather than survival efficiency. Blind fish living in dark caves evolved their eyes away, losing them completely through millions of years in lightless environments where vision provided no advantage. In completely dark cave systems where no light penetrates and vision cannot function, eyes represent a wasteful investment of energy and developmental resources. Fish ancestors entering these caves possessed functional eyes, but individuals with mutations causing eye degeneration suffered no survival disadvantage in the lightless environment. Meanwhile, the energy saved by not developing eyes could be invested in other systems like enhanced lateral line organs, sensing vibrations, or improved metabolic efficiency. Across countless generations, selection favored fish with reduced eye development, gradually eliminating functional eyes from cave populations. Over millions of years, the eyes degenerated completely, leaving only vestigial eye sockets filled with skin and bone, where eyes once existed. Genetic analysis reveals that cave fish retain genes controlling eye development. These genes simply remain inactive, suggesting the degeneration happened relatively recently in evolutionary time. If cave fish were somehow placed back into lit environments for millions of years, natural selection might eventually reinvigorate eye development, though this seems unlikely. This example demonstrates that evolution responds to current environmental conditions, maintaining traits that provide advantages and discarding traits that represent wasteful expenses. In darkness, eyes become evolutionary liabilities rather than assets, illustrating that there is no inherent drive toward complexity or capability in evolution. Opposable thumbs evolved separately in primates and marsupials, showing convergent evolution where unrelated lineages independently evolved the same adaptation, solving identical problems. Primates, including humans and our closest relatives like monkeys and apes, possess opposable thumbs, allowing precise grasping of small objects and manipulation of tools. Marsupials, a group of mammals fundamentally different from primates with different evolutionary history and geographic distribution, also independently evolved opposable thumbs in some species. Despite diverging from common ancestors over 100 million years ago and evolving on different continents, both lineages arrived at the same solution to the problem of precise object manipulation. This convergent evolution reveals that certain adaptations are so useful that multiple evolutionary pathways lead toward them independently. Convergent evolution occurs when different species facing similar environmental challenges or lifestyle requirements evolve similar solutions through natural selection, working on different genetic substrates. The genetic mechanisms controlling thumb development differ between primates and marsupials, yet both produce functionally similar opposable thumbs through distinct developmental pathways. This demonstrates that evolution is not constrained to a single solution for any given problem, but rather multiple solutions exist and can be discovered independently. The recurrence of similar adaptations across unrelated lineages suggests these adaptations solve fundamental problems encountered by many species in similar ecological contexts. Snakes lost their limbs through gradual evolution from lizard ancestors, with fossil intermediates and genetic remnants revealing the transition from four-legged to legless body plans. Modern snakes appear so distinct from their lizard relatives that their kinship might seem questionable, yet genetic evidence and fossil data confirm that snakes descended from lizard ancestors approximately 100 million years ago. Fossil intermediates show gradual reduction in limb size across evolutionary time, with transitional forms possessing small but functional limbs progressively reduced to vestigial structures. Modern snakes retain small pelvic bones and vestigial hip structures in their bodies, evolutionary remnants of the limbs their ancestors possessed. The loss of limbs provided evolutionary advantages in certain environments, allowing snakes to exploit ecological niches unavailable to four-legged creatures, particularly underground burrowing and movement through dense vegetation. Each intermediate stage in the transition from lizard to snake remained viable and successful in its environment, demonstrating that evolution proceeds through countless small modifications rather than sudden jumps. Genetic analysis reveals that snakes possess genes controlling limb development. These genes simply remain inactive or produce minimal structures compared to lizard relatives. The transition took millions of years, with thousands of intermediate forms existing between fully limbed ancestors and completely limbless descendants. This gradual transformation provides powerful evidence for evolution, revealing how major body plan changes can occur through accumulation of small modifications across immense time spans. The human hiccup reflex is an evolutionary relic from our fish ancestors with gills, representing an outdated reflex for clearing water from gills now purposeless, but retained because it causes no harm. When you hiccup, your diaphragm suddenly contracts involuntarily, creating a distinctive sound and sensation, though the reflex seems to serve no modern function. Evolutionarily, this reflex originated in fish ancestors possessing gills requiring water circulation for oxygen extraction. The hiccup reflex likely helped fish ancestors clear excess water from their gills when necessary, providing a survival advantage in those ancient environments. As fish evolved into tetrapods and eventually mammals, they abandoned gills in favor of lungs, yet the neural circuitry controlling the hiccup reflex persisted. Humans retained this ancient reflex because eliminating established neural circuits requires specific mutational changes, providing sufficient advantage to offset the energy cost of rewiring neural systems. Hiccups cause minor discomfort but no serious harm, providing no strong selection pressure favoring their elimination. This illustrates an important principle. Evolution doesn't erase unsuccessful adaptations from ancient ancestors, but rather preserves them if they do not actively harm survival or reproduction. Your hiccup represents a direct connection to fish ancestors living hundreds of millions of years ago, a reflexive echo of ancient evolutionary solutions preserved in modern nervous systems. Many aspects of human biology similarly represent evolutionary compromises where ancient structures persist alongside newer developments, creating sometimes inefficient but functional systems. Humans have wisdom teeth that no longer fit in modern jaws because our skulls and jaws evolved smaller over millions of years as our diet and lifestyle changed gradually. These third molars, appearing in late adolescence or early adulthood, represent teeth that served important functions for our distant ancestors with larger jaws and different dietary needs. Our ancestors consumed tougher, more fibrous foods requiring extensive grinding and crushing, necessitating larger teeth and more powerful jaw muscles. As human ancestors learned to cook food, making it softer and easier to process, the need for such massive grinding capacity diminished progressively. Over countless generations, natural selection favored individuals with smaller jaws because smaller skulls required less energy and resources to develop and maintain. The reduction in jaw size happened so rapidly in evolutionary terms that wisdom teeth, controlled by ancient genetic programming, continue developing even though space no longer exists for them. These vestigial teeth frequently become impacted, requiring surgical removal in modern humans, causing pain and infection that would have been serious problems for our ancestors. Yet wisdom teeth persist because evolution proceeds through modification of existing structures rather than complete redesign, and the disadvantage they cause is insufficient to drive their complete elimination. Your wisdom teeth carry testimony to the changing relationship between human biology and human lifestyle across millions of years of evolution. This mismatch between ancestral genetic programming and modern anatomy illustrates how evolution sometimes produces suboptimal outcomes when organisms change rapidly relative to evolutionary timescales. Some frogs evolved bright colors to warn predators of their poison, creating toxins specifically for defense through a process refining warning signals and toxin production simultaneously. Poison dart frogs display brilliant coloration in reds, blues, yellows, and other vivid hues that advertise their toxicity to potential predators. These colors serve no purpose for camouflage or thermoregulation, but rather function as warning signals, communicating danger to predators capable of learning associations between colors and bad experiences. The frogs evolved toxins in their skin that cause severe illness or death to predators attempting to consume them, making the warning signal truthful and meaningful. This coevolution demonstrates how two traits, warning coloration and toxin production, became linked through natural selection, favoring their simultaneous expression. Predators that learned to associate bright colors with toxicity survived and reproduced more frequently than those repeatedly poisoning themselves on toxic frogs. This created selection pressure, favoring frogs with the brightest, most recognizable warning colors, because these individuals suffered fewer predation attempts. Simultaneously, frogs with more potent toxins suffered fewer successful predation attempts, even from predators not fully familiar with warning signals. Over millions of years, this created an evolutionary arms race where warning coloration became progressively more vivid, while toxin potency increased correspondingly. The bright colors seem wasteful and conspicuous, yet they provide tremendous survival advantage by preventing predators from wasting energy on dangerous prey. This demonstrates that evolution sometimes favors traits appearing wasteful from a survival perspective, but providing advantages in different selective contexts. Polar bears evolved white fur from brown bear ancestors within the last 100,000 years, representing one of the fastest color adaptations in mammals driven by camouflage selection in Arctic environments. Polar bears and brown bears share relatively recent common ancestry, diverging only perhaps 600,000 years ago, making them closely related species still capable of producing viable hybrid offspring. Brown bears possess brown fur, providing excellent camouflage in forests and mountains where their ancestors evolved. Some brown bear populations gradually expanded into Arctic regions where brown coloration provided poor camouflage against snow and ice. In these white environments, individual bears with lighter fur coloration had significant hunting advantages, blending seamlessly into snowy landscapes while stalking seals. Across numerous generations, selection for lighter coloration intensified progressively, driving the gradual whitening of fur from brown toward lighter shades through intermediate stages. This color change occurred rapidly in evolutionary terms, with complete white coloration appearing within perhaps 100,000 years, a blink of an eye in geological time. The white coloration adaptation occurred so quickly that polar bear genomes retain many brown bear genes unchanged, revealing their recent divergence. The shift from brown to white involved not only coloration changes, but also physiological adaptations like thicker fur, different diet specialization, and behavioral modifications suited to Arctic life. This example demonstrates that evolution can produce dramatic adaptations remarkably quickly when strong selective pressures favor particular traits in isolated populations. Lactose tolerance in adult humans evolved only in the last 10,000 years in populations practicing dairy farming, representing one of the most recent human evolutionary adaptations. Before agriculture and dairy farming, human populations universally stopped producing lactase, the enzyme breaking down milk sugar after childhood weaning from breast milk. This pattern continues in most modern mammals, which naturally cease lactase production after infancy because milk consumption ceases. However, in populations originating from regions with long traditions of dairy cattle herding, particularly Northern Europe, the Middle East, and parts of Africa, individuals frequently retained lactase production into adulthood. This retention allowed adults to digest milk and consume dairy products, providing nutritional advantages during times of food scarcity. Genetic studies reveal that lactase persistence in adults resulted from mutations in regulatory regions controlling lactase gene expression, mutations that arose relatively recently in evolutionary time. These mutations spread rapidly through dairy farming populations because they provided significant survival and reproductive advantages in societies depending on dairy products. Lactose tolerance represents one of the clearest examples of cultural practices driving biological evolution, with agricultural innovation creating new selective pressures favoring previously disadvantageous traits. Most humans worldwide remain lactose intolerant as adults, reflecting the recent nature of dairy farming in human prehistory. This adaptation demonstrates that evolution operates on timescales measurable in thousands of years when selective pressures are sufficiently strong, a much faster rate than many evolutionary changes. Some plants evolved thorns and spines as defensive structures against herbivore grazing through millions of years of selection, favoring effective protection. Plants cannot flee from herbivores consuming their tissues, so they evolved chemical and physical defenses, discouraging herbivores from feeding extensively. Thorns and spines, sharp-pointed structures growing from stems and leaves, increase the cost of feeding by inflicting injury and discomfort on herbivores attempting to consume plant tissues. Some herbivores evolved toughened mouths and specialized feeding behaviors, allowing them to tolerate or avoid thorns while feeding, creating an evolutionary arms race. In response, some plants evolved progressively sharper, longer, or more densely distributed thorns through natural selection, favoring individuals with better defended tissues. This coevolution between plants and herbivores demonstrates how defensive and offensive traits can drive reciprocal evolutionary change, each adaptation triggering counter-adaptations in competing species. Desert plants frequently display particularly elaborate thorn defenses because water scarcity makes every plant more precious and worth defending extensively. Some herbivores evolved body armor or behavioral strategies specifically for dealing with thorny plants, like rolling into balls to protect soft tissues or careful feeding techniques, minimizing injury. Over millions of years, this ongoing competition between herbivores seeking nutrition and plants protecting tissues shaped dramatic morphological differences across plant species. The diversity of thorn sizes, shapes, and distributions reflects millions of years of evolutionary experimentation with defensive strategies and herbivore-driven selection pressures. The human spine contains remnants of a tail structure from our primate ancestors living in trees millions of years ago, preserved in the coccyx or tailbone. The coccyx consists of fused vertebrae at the base of the spine, representing the evolutionary remnant of a functional tail present in our distant primate ancestors. Primates using tails for balance while moving through tree canopies gained significant survival advantages, making tail presence beneficial in arboreal environments. As human ancestors gradually abandoned fully arboreal lifestyles and spent more time on the ground, the functional advantages of tails diminished progressively. Bipedalism, walking upright on two legs, altered spinal mechanics and made long tails less useful for balance compared to their utility in tree-dwelling primates. Over millions of years, natural selection progressively reduced tail length and complexity in human ancestors, eventually eliminating functional tails entirely while leaving only fused vertebral remnants. The coccyx serves no significant function in modern humans, though it provides minor attachment points for muscles and ligaments, representing an evolutionary compromise between complete elimination and retention. Some human embryos briefly show tail-like structures during fetal development before these structures regress, illustrating that evolutionary development echoes ancestral body plans. Occasionally, humans are born with vestigial tails, brief protrusions quickly removed surgically, demonstrating that genetic programming for tail development persists despite millions of years of selection against functional tails. Your tailbone connects you physically to tree-dwelling ancestors who depended on tails for survival in ancient forests. Chromosomal fusion events shaped human evolution, particularly chromosome 2 formed when two ancestral primate chromosomes fused together around 5 million years ago, creating a major reorganization. Modern humans possess 46 chromosomes, while our closest living relatives, chimpanzees and gorillas, possess 48 chromosomes spread across more chromosome structures. This difference arose when two ancestral chromosomes fused into a single, larger chromosome in early human ancestors, reducing chromosome count by 1. Evidence for this fusion appears in modern chromosome analysis, revealing that human chromosome 2 contains genetic sequences matching chimpanzee chromosomes 2A and 2B, placed end-to-end with fusion scars visible at the junction point. This chromosomal reorganization occurred in ancestral human populations perhaps 5 to 6 million years ago, when early hominins diverged from our last common ancestor with other apes. The fusion itself represented a significant genetic reorganization, but not necessarily an immediate functional change, as the same genetic information was preserved through the process of joining. However, this chromosomal rearrangement may have played a role in reproductive isolation, as individuals with different chromosome numbers sometimes produce sterile offspring when breeding with unmutated populations. Over millions of years following this fusion, humans continued evolving distinct characteristics while remaining genetically compatible within human populations carrying the fused chromosome. This chromosomal difference provides clear physical evidence distinguishing human genomes from our closest primate relatives and illustrates that evolution occurs through large-scale genetic reorganization, in addition to point mutations accumulating gradually. Plants evolved from algae that colonized land around 450 million years ago, requiring fundamental evolutionary innovations for terrestrial survival. Algae thrived in aquatic environments where water surrounded and supported their bodies, providing nutrients dissolved throughout, protecting tissues from desiccation. Land presented radically different challenges, requiring structural and physiological innovations, enabling survival in air where water is scarce and gravity acts unmediated by buoyancy. Early plants evolved root systems anchoring them in soil and extracting water and nutrients from terrestrial environments, replacing the osmotic absorption mechanisms of aquatic algae. They developed vascular tissues, transporting water and nutrients internally, replacing the diffusion systems sufficient in thin algal bodies. cell walls strengthened dramatically to provide structural support without water buoyancy, requiring cellulose and related compounds, creating rigid plant architecture. Protective outer layers called cuticles reduced water loss, allowing plants to prevent desiccation while maintaining gas exchange through specialized pores called stomata. The transition from aquatic to terrestrial life fundamentally reshaped plant evolution and Earth's ecosystems, enabling the eventual colonization of virtually every terrestrial habitat. Terrestrial plants gradually diversified into mosses, ferns, conifers, and eventually flowering plants, each group adding novel adaptations, refining terrestrial survival. This transition required thousands of simultaneous evolutionary innovations working in coordination, demonstrating how complex changes can be achieved through gradual accumulation of complementary adaptations over enormous time spans. Eyes evolved independently in at least 40 different animal lineages, showing that this adaptation was so advantageous that natural selection repeatedly invented eyes from scratch through independent evolutionary pathways. Despite the complexity of modern eyes, simpler visual systems appeared first in evolutionary history, gradually becoming more sophisticated as selection favored improved light detection and visual acuity. Some organisms possess simple light-sensitive cells providing only brightness detection. Others have lens-based systems focusing light, while others evolved compound eyes with thousands of separate optical units. The repeated independent origin of eyes across different animal groups demonstrates that natural selection discovered this adaptation multiple times because vision provides such tremendous survival and reproductive advantages. Each evolutionary lineage began with simple, light-sensitive cells, providing minimal directional information about light sources. Subsequent generations experiencing mutations, improving light detection, intensity, or directional sensitivity, reproduced more successfully, gradually refining visual systems. Some lineages accumulated genetic changes producing sophisticated camera-like eyes similar to human eyes, while others evolved entirely different designs like compound insect eyes, each optimized for particular ecological roles. The convergent evolution of eyes demonstrates that while evolution proceeds through random genetic mutations and differential reproduction, certain adaptations are so beneficial they recur repeatedly across independent lineages. Complex eyes appear unlikely to originate through random mutation, yet the gradual accumulation of small improvements from simple to sophisticated systems makes their evolution inevitable, given sufficient time and selective pressure. Photosynthesis evolved once in cyanobacteria and transformed Earth's atmosphere through millions of years of biological innovation, eventually enabling aerobic life and animal evolution. Ancient Earth possessed an atmosphere devoid of free oxygen, composed primarily of nitrogen and carbon dioxide with trace gases. Cyanobacteria evolved the capacity to capture light energy and split water molecules, using the energy to build sugars from carbon dioxide while releasing oxygen as a waste product. This photosynthetic innovation originated in a single microbial lineage billions of years ago, yet its consequences rippled through all subsequent Earth history. For millions of years, cyanobacteria continued evolving and diversifying while progressively releasing oxygen into the atmosphere through photosynthesis. This great oxidation event, beginning around 2.4 billion years ago, gradually transformed Earth's atmosphere from oxygen-free to oxygen-rich, fundamentally altering planetary chemistry. The rise of atmospheric oxygen represented an existential threat to anaerobic organisms dominating earlier Earth, driving numerous extinctions while creating opportunities for oxygen-respiring organisms. The evolution of aerobic respiration, allowing efficient energy extraction from organic molecules using oxygen, enabled far more complex and larger organisms than anaerobic metabolism permitted. Eventually, this oxygen-rich atmosphere enabled the evolution of eukaryotic cells, multicellular organisms, and ultimately animals, all depending ultimately on photosynthetic innovations developed by ancient cyanobacteria. A single evolutionary innovation in microbes, billions of years ago, set the stage for all subsequent life complexity, demonstrating how foundational evolutionary changes can cascade through planetary systems over inconceivable time spans. Dinosaurs ruled Earth for 165 million years before the asteroid impact ended their reign, a time span that far exceeds the entire history of mammals on this planet. When we consider the age of dinosaurs, we often think of brief dominance interrupted by sudden extinction, yet the reality tells a different story of remarkable stability. Dinosaurs appeared around 230 million years ago and diversified into thousands of species occupying nearly every ecological role, from tiny insectivores to massive herbivores and apex predators. For 165 million years, dinosaurs remained the dominant large animals on land, persisting through climate changes, continental drift, and countless smaller extinction events. This duration dwarfs the entire reign of mammals, which have existed for only around 200 million years and dominated large animal niches for far less time. Modern humans have existed for merely 300,000 years, a mere fraction of even mammalian history. The stability of dinosaur dominance suggests that this body plan and ecological strategy remained highly successful across immense time spans and varied environmental conditions. Only when an asteroid struck Earth 66 million years ago did this dominance end catastrophically, demonstrating that extinction often results from sudden environmental catastrophe rather than gradual, competitive replacement. The remarkable longevity of dinosaur dominance reveals that evolution can produce stable, successful systems persisting for hundreds of millions of years once established. This reminds us that complexity and diversity, once achieved, tend toward stability unless disrupted by external catastrophic events. Feathers originally evolved for insulation or display, not flight, thousands of generations before dinosaurs achieved actual aerial locomotion. Early dinosaurs possessed feathered bodies, yet their skeletal anatomy and body proportions indicate they remained fully terrestrial, incapable of powered flight. Paleontologists studying fossil evidence discovered that feathered dinosaurs retained small arms relative to body size and possessed heavy bodies unsuited for sustained flight. The presence of elaborate feathers on non-flying dinosaurs suggests that feathers provided functions other than flight, most likely insulation and visual display. Feathers provide excellent thermal insulation, trapping air that prevents heat loss, particularly valuable for active warm-blooded dinosaurs maintaining elevated body temperatures. Many early feathered dinosaurs displayed elaborate crests, colorful patterns, and elongated feathers suggesting sexual display and species recognition functions. Only after millions of years of feather evolution for these other purposes did some small dinosaur lineages eventually achieve powered flight using pre-existing feather structures. This represents a classic example of exaptation, where features evolve for one function later become co-opted for different purposes. The evolution of flight required not only feathered wings, but also skeletal modifications, reducing weight, changes in muscle attachment points, and neural adaptations enabling aerial maneuvering. The gradual transition from non-flying to flying dinosaurs demonstrates that major evolutionary transitions often build upon adaptations originally serving different purposes, requiring millions of years of refinement before achieving new capabilities. Tiktaalik, a 375-million-year-old fossil, shows the transition from fish to tetrapods, with both fish fins and tetrapod limbs representing a crucial intermediate form. This remarkable creature possessed anatomical features characteristic of both fish and terrestrial vertebrates, occupying an intermediate position in the evolutionary transition from aquatic to land-dwelling animals. Its skull structure closely resembled that of fish, yet its limbs showed tetrapod characteristics with bones homologous to modern amphibian and mammalian limbs. The fins of Tiktaalik contained internal skeletal elements resembling early tetrapod limbs, suggesting these structures functioned as both fins and primitive legs. Tiktaalik likely inhabited shallow water environments, using its fin limbs to propel itself through water and occasionally haul itself onto muddy banks. Its body shape and size would have made it poorly suited to sustained terrestrial locomotion, indicating it remained primarily aquatic. The discovery of Tiktaalik and similar transitional fossils provided powerful evidence supporting evolutionary theory by demonstrating intermediate forms between major animal groups. Paleontologists predicted where transitional fossils should appear in rock strata based on evolutionary theory, then discovered Tiktaalik in precisely predicted geological formations. This fossil reminds us that evolution proceeds through countless intermediate stages, each viable and successful in its environment, gradually accumulating changes that eventually produce radically different body plans. The existence of forms like Tiktaalik demonstrates that major evolutionary transitions leave fossil evidence revealing the gradual process of transformation. Whales communicate using low-frequency songs that travel vast ocean distances, evolved for long-distance mating calls across featureless marine environments where vision fails. Whales produce vocalizations ranging from simple clicks and whistles to complex songs lasting hours, with different whale species possessing distinct dialects and call types. Humpback whales produce elaborate songs that change over seasons, with populations learning new song patterns from neighboring groups through cultural transmission. The low-frequency calls of baleen whales travel enormous distances through water, potentially reaching other whales hundreds of kilometers away in the ocean depths. These vocalizations evolved to enable communication in marine environments where light rarely penetrates beyond shallow depths and visual signaling becomes impossible. The frequency of whale songs represents a sound optimization for underwater propagation, as these frequencies travel farther through seawater than higher frequencies. Male whales use songs for mating advertisement, establishing territories, and attracting females, functions explaining the evolution of increasingly elaborate and energetically expensive vocalizations. Females likely evolved preferences for complex songs, creating sexual selection pressure favoring males producing the most impressive displays. Different whale populations maintain distinct song dialects passed across generations through cultural learning, suggesting sophisticated vocal learning abilities evolved in whale ancestors. The sophistication of whale communication demonstrates that evolution produces complex behaviors and sensory systems finely tuned to organisms' ecological niches and environmental challenges. Some bacteria developed flagella, whip-like structures for movement, through evolutionary assembly of simpler components into integrated nanomachines. Bacterial flagella represent extraordinarily complex structures, consisting of dozens of protein components assembled into rotating motor systems propelling bacteria through liquid environments. The flagellum consists of a hook connecting the motor to a long, thin filament generating propulsion through rotation, with the entire structure powered by electrochemical gradients across the bacterial cell membrane. This rotary motor operates at rotational speeds exceeding 100,000 revolutions per minute, demonstrating remarkable engineering efficiency at the molecular scale. The evolutionary origins of flagella remain debated, but evidence suggests they assembled gradually from pre-existing cellular components through gene duplication and modification. Simpler protein structures controlling cell movement in some bacteria likely provided the ancestral starting point for flagellar evolution. Genes controlling flagellar proteins show similarity to genes controlling other cellular structures, suggesting they arose through modification of existing genetic material rather than sudden invention. The gradual assembly of flagellar components over millions of years produced increasingly functional movement systems, each intermediate stage viable and potentially beneficial. Contemporary bacteria reveal varying flagellar complexity, suggesting different stages of flagellar evolution persist in modern species. The evolution of flagella illustrates how natural selection can assemble extraordinary complexity from pre-existing components through millions of years of incremental modification and refinement. Plant seeds evolved protective coats and stored nutrients, allowing them to survive dormancy and colonize drier habitats and seasonal environments. Early plants reproduce through spores dispersed by wind, similar to modern ferns and mosses, yet spores lack protective structures and stored energy reserves, limiting survival. The evolution of seeds represented a major innovation, encasing reproductive material in protective tissue and providing nutrient reserves, enabling germination in harsh conditions. The seed coat protects the embryonic plant from physical damage and desiccation, allowing seeds to survive dry periods lethal to unprotected spores. Nutrient stores within seeds, called endosperm, provide energy, enabling the young plant to grow roots and establish itself before photosynthesis begins sustaining it. This combination of protection and stored nutrients enabled plants to colonize seasonally dry environments and survive extended dormancy periods. Some seeds evolved elaborate dispersal mechanisms, including wings for wind dispersal and fleshy fruits attracting animals who spread seeds over distances. Seeds represent such an effective innovation that seed-bearing plants eventually dominated terrestrial ecosystems, replacing earlier plant groups dependent on spores. The diversity of seed structures reflects millions of years of evolution, optimizing strategies for survival, dormancy, and dispersal across varied environments. This innovation demonstrates how a single evolutionary development, refined across millions of years, can fundamentally restructure entire ecosystems. Flowers evolve specific shapes and colors matching pollinator vision and preferences, creating intricate partnerships refined through millions of years of coevolution. Flowering plants evolved petals in various colors, each color possibly attracting different pollinator groups based on their visual capabilities and preferences. Bees possess color vision, centered in the ultraviolet, blue, and yellow ranges, while butterflies see different color spectrums, and birds perceive red and orange particularly effectively. Flowers evolved colors matching their primary pollinators' visual sensitivities, suggesting that pollinator vision shaped flower color evolution through selection pressure favoring flowers visible to their preferred pollinator species. Beyond color, flowers evolved specific shapes optimizing pollinator access to nectar and pollen while ensuring pollen deposition on pollinator bodies. Some flowers evolved narrow tubes, attracting only specialized pollinators, preventing less effective pollinators from accessing nectar while ensuring targeted pollen transfer. Bees see ultraviolet patterns invisible to human eyes, yet visible on many flowers as landing strips guiding them to nectar sources. The intricate matching of flower morphology to pollinator anatomy and behavior demonstrates coevolution, where two species traits evolved reciprocally through mutual selective pressures. Flowers without specialized pollinators produce copious pollen, relying on wind dispersal, a less efficient strategy requiring enormous pollen production. The evolution of specialized pollination relationships represents millions of years of reciprocal adaptation, each innovation in one species triggering counter-innovations in the other. Primates evolved forward-facing eyes for improved depth perception in forest canopies, an adaptation providing tremendous advantages for tree-dwelling ancestors. Most mammals possess eyes positioned on the sides of their heads, providing panoramic vision useful for detecting predators approaching from any direction. Primates evolved forward-facing eyes positioned on the front of the head, reducing peripheral vision but enabling stereoscopic vision through overlapping fields. Stereoscopic vision creates three-dimensional depth perception, allowing precise distance judgment essential for judging branches while leaping through forest canopies. Tree-dwelling ancestors navigating complex three-dimensional environments gained tremendous survival advantages through accurate depth judgment, reducing misjudged jumps resulting in fatal falls. The forward-facing eye position required evolving different orbital bone structures and facial anatomy, significant skeletal modifications reflecting the importance of this adaptation. The visual cortex processing stereoscopic information expanded significantly in primate brains, enabling sophisticated spatial perception and coordination. Modern primates retain this forward-facing eye configuration despite ancestral tree dwelling only partially applying to many species, suggesting the depth perception advantages remain valuable. The evolution of forward-facing eyes illustrates how specific anatomical adaptations target organisms' ecological challenges, with selective pressures favoring traits directly improving survival in particular environments. The extensive neural expansion supporting stereoscopic vision in primate brains may have provided foundations for later evolution of complex visual processing, enabling tool use and abstract thought. The immune system evolved elaborate recognition mechanisms distinguishing self from non-self, developing sophisticated defense systems perfected over hundreds of millions of years of pathogen exposure. Early life forms possessed only innate immune responses, relying on general mechanisms attacking unfamiliar molecules without specific recognition of particular pathogens. As animals became more complex, pathogens became more varied and sophisticated, creating selection pressure favoring immune systems with greater specificity and learning capacity. Adaptive immunity evolved, enabling organisms to recognize specific pathogens and mount targeted responses, with immune cells retaining information about previous infections. Antibodies, proteins specifically binding particular pathogens, represent evolved recognition molecules refined through millions of years of coevolution between hosts and parasites. The immune system's capacity to recognize vast numbers of potential pathogens suggests evolution-produced mechanisms generating enormous molecular diversity, enabling recognition of novel pathogens. Each individual's immune cells develop a unique recognition repertoire before birth, creating different recognition patterns across human populations and improving collective pathogen resistance. The evolution of immune system sophistication demonstrates how coevolution between hosts and pathogens drives reciprocal adaptation, each innovation in pathogen evasion triggering counter-innovations in host immunity. Cancer, aging, and autoimmune diseases result partly from trade-offs in immune system evolution, showing that no adaptation solves all biological problems perfectly. The immune system reminds us that evolution produces compromises, reflecting complex interplay among competing selective pressures. Hair and fur evolved as insulation mechanisms in mammalian ancestors around 200 million years ago, enabling warm-blooded animals to remain active across diverse climates and times. Early reptilian ancestors possessed scales providing structural protection but minimal insulation, adequate for cold-blooded animals with body temperatures matching environments. Mammals evolved warm-bloodedness, maintaining elevated body temperatures through metabolic heat production, requiring mechanisms reducing heat loss to environments. Hair and fur provide excellent insulation by trapping air, creating dead space, reducing heat transfer from skin to environment. The evolution of fur coincided with development of metabolic machinery, enabling sustained heat production, both innovations supporting warm-blooded biology. This thermal adaptation enabled mammals to remain active during cold periods and in cold climates, expanding available ecological niches and allowing nocturnal activity. Different mammals evolved fur of varying thickness, density, and length, with desert mammals developing thin, sparse fur, while Arctic mammals evolved dense, thick coats. Seasonal fur changes in some mammals represent evolved responses to environmental temperature variation, optimizing insulation seasonally. Hair serves multiple functions beyond insulation, including sensory perception through specialized follicle receptors and chemical communication through scent gland secretion. The evolution of fur represents millions of years of refinement, producing efficient insulation systems, matching diverse environmental conditions across mammalian distributions. Echolocation evolved independently in bats and dolphins, demonstrating convergent sensory adaptation where two mammalian lineages solved the darkness problem through acoustic evolution. Both bats and dolphins inhabit environments where vision provides limited or no advantage, yet both require precise spatial awareness for hunting and navigation. Bats evolved the capacity to produce high-frequency sounds, listen to returning echoes, and translate these acoustic signals into detailed spatial maps guiding flight. Dolphins independently evolved similar sonar capabilities using different frequencies and neural processing, yet achieving comparable spatial resolution and hunting precision. The neural systems processing echolocation information expanded significantly in both lineages, creating dedicated brain regions for acoustic analysis unavailable in most mammals. This parallel evolution of complex sensory systems demonstrates that certain adaptive solutions, while extraordinarily sophisticated, recur repeatedly when environmental challenges are sufficiently similar. The convergent evolution of echolocation suggests that natural selection discovers this adaptation independently whenever organisms occupy dark environments requiring precise navigation and hunting. Each species' echolocation system reflects unique evolutionary history and specific ecological demands, yet both achieve functional equivalence through different neural and anatomical implementations. The sophistication of echolocation, allowing detection of objects millimeters in size and tracking of rapidly moving prey, represents one of evolution's most remarkable sensory innovations. Bats and dolphins remind us that evolution frequently produces equivalent solutions to similar problems through independent pathways, a testament to natural selection's power in discovering optimal adaptations. Camouflage patterns in moths and caterpillars show ongoing evolution, matching their environmental backgrounds through continuous refinement by predation pressure. Peppered moths display coloration patterns closely matching the bark surfaces, where they rest during daylight, reducing visibility to bird predators hunting by sight. Different moth populations inhabiting different tree species show corresponding color variations, with moths matching their local bark colors more closely than moth populations from distant regions. This geographic variation reflects ongoing evolution where local predation pressure selects for camouflage, matching local environmental conditions. Caterpillars evolved camouflage patterns matching their host plants, with some appearing as bird droppings, others resembling twigs, and still others displaying leaf-like coloration. The diversity of camouflage strategies among caterpillars reflects millions of years of evolution, optimizing concealment against diverse predators and varied host plant appearances. Some caterpillars evolved color changes, matching seasonal changes in host plant appearance, representing behavioral evolution, complementing morphological adaptation. Predators exert constant selection pressure, favoring better camouflaged individuals, driving continuous refinement of camouflage patterns across generations. As predator eyesight evolved, becoming more acute, moths and caterpillars required progressively more effective camouflage, maintaining an evolutionary arms race between concealment and detection. The continuous fine-tuning of camouflage patterns demonstrates that evolution is ongoing and responsive, constantly adjusting populations to match environmental conditions and predator capabilities. Venom glands evolved multiple times in different animal lineages from modified salivary glands, suggesting they evolved from common glandular ancestors through convergent evolution. Snake venom, insect venom, and spider venom all share surprising chemical similarity, suggesting they evolved from similar ancestral gland structures through independent modification. Salivary glands produce lubricating secretions facilitating food manipulation and initial digestion, present in most animals and representing ancient, well-established glandular structures. Some lineages evolved mutations altering salivary gland secretions to produce toxic compounds, initially perhaps inadvertent byproducts that eventually provided selective advantages. Animals producing toxic secretions suffered fewer predation attempts and succeeded better in prey capture, selecting for mutations, increasing venom potency and production. Over millions of years, selection refined venom composition, toxin concentration, and venom delivery mechanisms, producing highly specialized venomous systems. The chemical convergence among venoms from different lineages suggests that certain toxins represent particularly effective solutions to predation problems or prey immobilization. Venom evolution demonstrates that major innovations frequently build upon pre-existing structures modified through relatively few genetic changes rather than requiring entirely novel structures. The repeated evolution of venom systems across independent lineages suggests this innovation provides such substantial advantages that natural selection repeatedly discovers it. Contemporary venomous animals display enormous diversity in venom composition and delivery systems, reflecting millions of years of independent evolution, optimizing venoms for specific predators and prey. Ears evolved from fish jaw bones, repurposed for hearing in early mammals, transforming gill support structures into sophisticated sound-conducting mechanisms. The bones in your middle ear, the hammer, anvil, and stirrup, originated as gill support structures in ancient fish ancestors. These bones function to stabilize gills and facilitate water flow through gill chambers, essential for aquatic respiration. As fish evolved into tetrapods and eventually mammals, the transition from gill breathing to lung breathing made these bones unnecessary for their original function. Early mammals retained these bones in modified form, yet remained non-functional for hearing, positioned where they had always been in the jaw joint region. Over millions of years, selection favored anatomical modifications that repositioned these bones to conduct sound vibrations from the eardrum to the inner ear. The hammer bone, originally a jaw support called the articular bone, became disconnected from the jaw and repositioned to vibrate with the eardrum. The anvil and stirrup bones underwent similar modifications, eventually creating a three-bone chain efficiently transmitting sound vibrations to the fluid-filled inner ear. This transformation required extensive skeletal reorganization and neural rewiring, yet provided such substantial hearing advantages that natural selection strongly favored these changes. Modern mammals process sound far more effectively than reptiles, owing partly to this evolved hearing system constructed from repurposed ancient structures. The evolution of ears demonstrates that evolution frequently transforms obsolete structures into new functions through gradual modification over millions of years. Social behavior in ants evolved through kin selection and colony-level adaptations, creating societies where individual workers sacrifice reproduction for colony success. Ants represent one of evolution's most remarkable social achievements, with colonies containing thousands or millions of individuals coordinating behavior with striking precision. Worker ants sacrifice individual reproduction, dedicating their lives to caring for the colony's queen and her offspring, representing a profound evolutionary paradox since natural selection should favor reproduction. This apparent paradox resolves through kin selection, a mechanism where individuals gain inclusive fitness by helping close relatives reproduce. Worker ants share genetic material with their queen and nestmates, so assisting their reproduction propagates shared genes despite workers not reproducing personally. The evolution of eusocial behavior, with sterile workers supporting reproduction in others, represents one of evolution's most complex adaptations, requiring millions of years of refinement. Early ant ancestors likely showed facultative sociality, where individuals sometimes helped raising siblings before reproducing themselves. Gradual behavioral modifications eventually produced obligate sterile castes, with workers never reproducing, pure specialists in colony labor. division of labor evolved among workers with different sizes and morphologies specialized for different tasks, optimizing colony efficiency. Colony-level selection may have reinforced these behaviors, with colonies showing better cooperation, outcompeting less cooperative rivals. The evolution of ant societies demonstrates that natural selection operates at multiple biological levels simultaneously, from genes to individuals to populations to colonies. Placental development in mammals evolved gradually from egg-laying ancestors, representing the most sophisticated reproductive strategy among vertebrates. Ancient mammal ancestors laid eggs, similar to modern monotremes like platypuses and echidnas, with embryos developing externally. Intermediate mammalian lineages retained eggs internally, incubating them inside the mother's body while providing no nutritional support beyond yolk reserves. Early marsupials evolved brief internal development, followed by birth of underdeveloped young, continuing growth in pouches, supplemented by milk production. Placental mammals evolved the most elaborate system, retaining embryos internally while creating sophisticated nutrient transfer systems through the placenta. The placenta represents an extraordinary adaptation, allowing maternal blood and fetal blood to approach closely without mixing, enabling efficient nutrient transfer and waste removal. Placental development allows embryos to grow to relatively large size before birth, producing more mature offspring, better equipped for survival than marsupial infants. The evolution of increasingly complex pregnancy systems coincided with evolution of mammary glands, creating another major innovation enabling offspring nutrition post-birth. Placental mammals achieved reproductive success through longer pregnancy and smaller litter sizes compared to egg-laying mammals, representing a different evolutionary strategy optimizing different aspects of reproduction. The gradual evolution from egg-laying to placental reproduction demonstrates that major reproductive innovations emerged through millions of years of incremental modification, each stage viable and successful in its ecological context. Hemoglobin protein structure remains nearly identical across species separated by billions of years, showing that successful solutions, once evolved, persist relatively unchanged when they continue functioning optimally. Hemoglobin, the protein carrying oxygen in blood, displays essentially identical structure across humans, horses, fish, and other vertebrates despite billions of years of evolution separating these species. The similarity extends to amino acid sequences, with hemoglobin from distantly related species differing in only a few positions among hundreds of amino acids. This molecular conservation suggests that hemoglobin structure represents an optimal solution to oxygen transport, requiring little modification across vast evolutionary time. Once evolution discovered this solution billions of years ago, subsequent modifications offered minimal improvement in oxygen transport efficiency, removing selective pressure for change. Genes encoding hemoglobin have accumulated mutations randomly across billions of years, yet most mutations altered non-critical regions while maintaining core functional structure. The rare mutations significantly altering hemoglobin function typically reduce fitness, generating selection against these changes and maintaining hemoglobin stability. This molecular stasis demonstrates an important principle. Evolution does not continuously change structures, but rather modifies them when selective pressures drive change and preserves them when they function optimally. Different species show slight hemoglobin variations reflecting unique evolutionary modifications, yet these variations represent minor elaborations on an ancient, highly successful design. Hemoglobin reminds us that evolution balances innovation with conservation, exploring new solutions when circumstances demand while preserving ancient successes when they continue serving effectively. Some organisms evolved extreme heat or cold tolerance through specialized proteins and physiological adaptations, enabling survival in lethal temperatures. Desert insects survive environmental temperatures exceeding 50 degrees Celsius through heat-resistant proteins maintaining structural integrity despite thermal stress. Arctic fish survive water temperatures below 0 Celsius through antifreeze proteins lowering their blood freezing point below environmental temperatures. These extreme temperature tolerances required evolution of specialized protein structures more stable at temperature extremes than normal proteins. Heat-tolerant proteins fold into configurations resisting unfolding despite thermal vibrations that would denature ordinary proteins. Cold-tolerant organisms evolved antifreeze compounds, reducing ice nucleation, allowing body fluids to remain liquid at temperatures where normal water would freeze solid. The biochemistry of temperature tolerance illustrates how evolution produces specialized solutions to extreme environmental challenges through molecular innovation. Organisms inhabiting extreme temperature environments underwent rapid evolution of temperature tolerance traits when populations colonized these habitats from moderate climate origins. The repeated evolution of heat tolerance in desert organisms and cold tolerance in Arctic species demonstrates that evolution discovers similar solutions independently when environmental challenges are sufficiently similar. Extreme temperature tolerance represents thousands of simultaneous genetic modifications, refining proteins and physiological systems, demonstrating that evolution can produce complex adaptations through accumulation of numerous small changes. These specialized adaptations remind us that evolution produces extraordinary diversity of solutions, enabling life in environments superficially appearing uninhabitable. Color patterns evolved through natural selection for camouflage in backgrounds, with each pattern representing evolved visual disruption, camouflaging prey from predators searching for recognizable shapes. Leopard spots, zebra stripes, and tree bark patterns each represent evolved camouflage designs refined through millions of years of predator-prey interactions. Spots and stripes disrupt visual perception of animal outlines, making individual animals difficult to distinguish from background environments. Different habitats selected for different pattern types, with open grassland predators favoring stripes, while forest predators selected for spotted patterns. Predators evolved visual perception systems detecting patterns and shapes, driving counter-evolution of increasingly effective camouflage, breaking up recognizable silhouettes. Some patterns evolved for purposes beyond camouflage, including species recognition and sexual display, creating complex selective pressures. The diversity of coloration patterns reflects millions of species independently evolving appearance, matching their respective habitats. Contemporary populations still show geographic variation in coloration patterns, with regional variants better matching local backgrounds than distant variants. This ongoing evolution of coloration patterns demonstrates that natural selection continuously refines appearance. Matching environmental conditions. The intricate camouflage patterns observed across animal diversity represent countless millions of years of predation, pressure shaping appearance toward ever better concealment. Sleep patterns evolved differently across species based on predation risk and environmental demands, creating dramatic variation in sleep duration and intensity. Prey animals, constantly vulnerable to predation, evolved light sleep with frequent awakenings enabling rapid escape responses when predators attack. Herbivores like giraffes and zebras sleep only a few hours daily, maintaining constant vigilance for predators. Predators, relatively secure once feeding, evolved deeper sleep and longer sleep durations, allowing extended rest without immediate survival risk. Humans show intermediate sleep patterns reflecting our ancestors' position as both predators and prey, with sufficient sleep for tissue repair, yet enough vigilance for safety. Sleep duration varies dramatically among species based on predation risk and metabolic demands, showing that sleep itself evolved as an adaptation. Dolphins evolved uni hemispheric sleep, with only half the brain sleeping while the other half maintains vigilance, enabling both rest and predator detection simultaneously. This remarkable adaptation allows dolphins to rest while remaining alert for threats and maintaining voluntary breathing. The evolution of different sleep strategies demonstrates that natural selection shapes even apparently universal functions like sleep based on species-specific ecological circumstances. Sleep architecture and duration reflect evolutionary trade-offs between tissue maintenance requirements and safety considerations. Predation risk appears to be one of the strongest selective forces shaping sleep evolution across diverse animal species. Smell and taste receptors evolved as chemical detection systems for identifying food, mates, and threats, representing ancient sensory systems predating vision and hearing. Chemoreceptors, the molecular basis of smell and taste, evolved billions of years ago in early organisms detecting chemicals in their environment. These ancient sensory systems enabled microorganisms to locate nutrients and avoid toxins, providing fundamental survival capabilities. As animals became more complex, chemoreceptory systems elaborated with thousands of different receptor types, enabling detection of vast chemical diversity. Vertebrates evolved specialized olfactory receptors detecting airborne chemicals in mammals, and aquatic organisms detecting dissolved chemicals. Taste receptors evolved specialized systems detecting food quality, salinity, and toxins, enabling rapid assessment of food safety. The complexity of olfactory systems rivals or exceeds visual system sophistication, with dogs possessing millions of olfactory receptors enabling detection of scents imperceptible to humans. Chemical communication between organisms evolved based on these chemoreceptory systems, with pheromones enabling mate attraction and chemical warnings of danger. Sexual selection favored enhanced olfactory capabilities in some species, driving evolution of increasingly sensitive chemical detection. The antiquity of chemical senses, predating vision by billions of years, demonstrates that evolution proceeds through gradual elaboration of ancient sensory foundations. Smell and taste remind us that the sensory capabilities we take for granted represent billions of years of evolutionary refinement and specialization. Some organisms evolved reproduction strategies that vary between asexual and sexual methods, each offering different advantages in particular environmental circumstances. Asexual reproduction produces genetically identical offspring through simple cell division or fragmentation, allowing rapid population growth when conditions favor existing traits. Sexual reproduction mixes genetic material from two parents, producing offspring with novel genetic combinations potentially better suited to changing environments. Some organisms evolved the flexibility to reproduce sexually when genetic diversity offers advantages, then revert to asexual reproduction when rapid growth becomes advantageous. Plants frequently employ both strategies simultaneously, reproducing sexually through seeds while also spreading through runners or fragmentation. This reproductive flexibility demonstrates that evolution does not commit populations to single strategies, but rather produces mechanisms enabling adaptation to changing circumstances. Sexual reproduction carries costs, including the energy required to find mates and the dilution of parental genes in offspring, yet offers benefits through genetic diversity. Asexual reproduction preserves advantageous genetic combinations, yet risks extinction through inability to adapt to changing environments. The coexistence of both reproductive strategies across diverse organisms shows that evolution has not selected one strategy as universally superior, but rather maintains both. The prevalence of sexual reproduction in complex organisms suggests that genetic diversity becomes increasingly important as organisms become more complex and face more varied environmental challenges. Photosynthetic organisms convert sunlight into chemical energy through evolved light-capturing molecules refined across billions of years of chemical evolution. Chlorophyll and related pigments absorb specific wavelengths of light, representing billions of years of chemical evolution in capturing solar energy. These pigments evolve to absorb light wavelengths abundant in sunlight while reflecting other wavelengths, optimizing energy capture. Different photosynthetic organisms evolved slightly different pigment compositions, allowing them to exploit different light environments and wavelengths. Cyanobacteria evolved early photosynthetic systems billions of years ago, establishing fundamental mechanisms that all subsequent photosynthetic organisms refined. The efficiency of photosynthetic energy capture rivals or exceeds human-engineered solar panels, demonstrating that evolution produced remarkably efficient energy conversion systems. Pigment molecules work in coordination with protein complexes, forming reaction centers where light energy converts to chemical bonds. The complexity of photosynthetic machinery suggests millions of components evolved simultaneously through natural selection. Different photosynthetic pathways evolved with C3, C4, and Km pathways representing different solutions to photosynthetic efficiency in varied environmental conditions. The diversity of photosynthetic mechanisms reflects evolution's repeated refinement of light capture and energy conversion across billions of years. Parasites co-evolved with hosts, each shaping the other's survival strategies through millions of years of intimate antagonistic relationships. Tapeworms that infect intestines evolved simplified body plans, losing organs unnecessary in hosts, adapting to internal parasitic life. Host organisms evolved immune defenses specifically targeting parasites, creating selection pressure favoring parasite evasion mechanisms. Some parasites evolved transparency or mimicry of host tissues, avoiding immune detection through camouflage at molecular levels. Others evolved rapid mutation rates, generating diversity in surface molecules, staying ahead of immune system recognition. This ongoing coevolution between hosts and parasites creates evolutionary arms races where each innovation in one species triggers counter-innovations in the other. Parasite populations exert strong selection on host immune systems, driving rapid evolution of immune recognition capabilities. Hosts evolved behavioral responses to parasitism, including grooming behaviors, removing parasites, and dietary preferences toward plants with antimicrobial properties. The intimate relationship between parasites and hosts demonstrates that evolution operates through interaction between species not in isolation. Contemporary parasite-host interactions reveal active, ongoing coevolution, with parasite populations continuously adapting to host defenses while hosts evolve new immune responses. This endless cycle of coevolutionary change demonstrates that evolution generates complexity through competition between organisms sharing intimate ecological relationships.